And with this, we'll close with our last speaker, Dr. Vanessa Milanese Holanda, who also couldn't be here, unrelated to why Tobias couldn't be here. And Vanessa is a functional neurosurgeon in Brazil and adjunct assistant professor of neurosurgery at the Mayo Clinic in Jacksonville, Florida. And she will talk today about deciphering deep brain networks and functional neuroanatomy.
Hello, everybody. Please receive my warm regards directly from Austria. Unfortunately, due to a schedule conflict, I was not able to be there in person. But I'd like to thank Sarah, Jonathan, and Boris, and all the organizers to put together this amazing session. And then I'd like to first introduce myself. I'm Vanessa Milanese, a functional neurosurgeon from Brazil. I work at the Deficiência Portuguesa de São Paulo, and I'm also a director at Nomo Clinic, São Paulo, Brazil. And I'm also adjunct faculty at the Mayo Clinic, Florida. And I got elected I'm the president elected for the Brazilian Society for Stereotactic and Functional Neurosurgery. So I'm going to present to you from pathways to purpose, decoding deep brain networks. And beyond all the technology evolution that we have in our hands for targeting, it is very important to understand all the neuroanatomy involved in each procedure that we perform. You may think about the stereotactic procedures, such as lesions or deep brain stimulation. But now I want to invite you to have an immersive experience and to have, as my mentor would usually say, Dr. Rotten, to have the X-ray view. And when the patient first comes to the hospital, we implant this arc we numb the area, and then we implant this arc. We have the CT to fuse with the MRI, and then we put the YZ coordinates to reach the first. Here I'm going to mention to you about the GPI targeting, and usually I'm using a very like FGARE sequence that it was built at University of Florida, that we can really see the GPI and GPE and get to these deep structures. So I want to invite you to go and understand every step of this procedure, every structure that we cross until reach the GPI. So first we have the superior frontal gyrus, and then we have the U fibers. And once we go deeper, we are going to get the superior longitudinal fasciculus. I have a little bit here that was removed. And then we have the corona radiata. And then we have, going deeper, the putamen, globus pallidus externus, globus pallidus internus, and right inferior to it, we have the optic tract. That is a very important landmark for us when we are performing GPI DBS. Here in this anterior view, in one of my dissections, we have here both sides the cingulum, and then we have the optic tract here, both sides. And then I removed a portion of those fibers anteriorly, and we have the anterior commissure. And then going more posteriorly, we have GPI here, right superior to the optic tract, both sides. And then I got to show some of the connections of the GPI. We have connections with the thalamus, substantia nigra, pedunculopontine nucleus, globus pallidus externus, subthalamic nucleus, amygdala, and putamen. And once we implant and get to stimulate the specific areas, and mainly connected to the supplementary moral area and the supplementary moral cortex, we get to improve a lot of these dystonic movements, as you can see here in this patient. So this is something that it's really life-changing for them. So moving to the subthalamic nucleus, we have here in this lateral view also, we're going to cross the superior frontal gyrus. And then once we go deeper, we cross the U fibers, superior longitudinal fasciculus, corona radiata, until reach the thalamus, and then the zona incerta that it's right inferior. So here we have the thalamus, right inferior to it we have zona incerta, and right inferior to it we have subthalamic nucleus. In the superior oblique view, we have the red nucleus, superior, anterior, and lateral. To it, we have the STN here, and usually we get the lateral and posterior portion of the STN when we are simulating. And then we have here the posterior limb of the internal capsule, anterior limb of the internal capsule, and we have here GPI, GPE, and the putamen. And anteriorly here we have the caudate, and we have these fibers separating the head of the caudate, and the putamen is are fibers of the anterior limb of the internal capsule. And here in this lateral view, once we go deeper, we have here the optic tract getting to the lateral geniculate body. We have these fibers going up here is the corticospinal tract. We have some fibers going up of the medial lemniscus, and then we have here red nucleus. Right here is the pedunculum pontine nucleus, superior colliculus, inferior colliculus, lateral geniculate body, and then we have the STN here, and we can even see some connections here in this picture between the STN and the mammillary body, and between the STN and the hypothalamus, and between the STN and the olfactory tubercle. Here's an important landmark that it's the anterior commissure. So here I made a cut, and then we have portion of the putamen that continues with the substantia nominata, and posterior to the anterior commissure, we have GPE and GPI here. And then I got to show some of these connections. We have connections with the caudate nucleus, medial frontal cortex, pedunculum pontine nucleus, substantia nominata that might explain why some patients have some limbic change when we are stimulating anteriorly during the STN, globus pallidus internus, substantia nigra, olfactory tobacco, hypothalamus that might explain why some patients have some dysautonomia when we are stimulating anteriorly there in the STN, mammillary bodies, some problems with memory. Remember, mammillary body, memory, and fornix also, and also the putamen. So if you want to learn more and see, you can just get your cell phone and get in this QR code the paper. And I highly recommend you to also read the supplementary data here. And like going a step forward, I got to contribute with Dr. Andreas Horn and his amazing team in Harvard. So they perform these deep brain simulation of symptom-specific networks in Parkinson's disease. And we got to show that once we are simulating specific fibers, in this, let's say a patient that has a lot of tremors, once we stimulate the fibers that are connected to M1, we get to improve tremor. When we stimulate the areas that are connected to the supplementary motor area, we get to improve bradykinesia and rigidity, for example. And we also have specific fibers for axial symptoms. And in this paper, I also highly recommend you to get to the supplementary data. You can see amazing atlas there that Nandita and Helen Frederick put together. And you can see many of my dissections also there to help to understand and to validate those small tracts. And talking about these small tracts, we have some of these, we can really see here some of the connections between the STN, GPI, GPE, and also the areas of the fibers. So we have this color coding for the fibers that are connected to the supplementary motor area, to the primary motor cortex, as I mentioned, like the M1. You can see a lot of green here, and also some of the axial symptoms that might help, and also in the premotor pre-supplementary moral area, we can get to improve also rigidity and also some axial symptoms. So with that, and this is a very small change that I'm doing in my targeting. So for this patient that he had a lot of tremor, I went just 0.5 millimeter posteriorly to the area that I'm usually going, and then I got to stimulate those fibers connected to M1, and he improved a lot his tremors. So at the end, it's very helpful to help those patients. So moving to the thalamus, the last target that is also very important to reduce tremors. In this lateral view, I got to remove most of the fibers of the cortex and reaching the thalamus. We have here the mammillary body, getting the mammillothalamic tract, getting to the anterior nucleus of the thalamus. We have some fibers coming up here of the medial lemniscus, getting to VC or VP, and we have some fibers crossing here of the dentate, dentatobothalamic tract, getting to the VIM here, and then anteriorly here we have the VO. So usually the area that we stimulate, it's right here between the VO and VIM to improve tremors. So here you can see the dentate in this anterior view. We have these fibers crossing and getting here to the VIM. We also have some small portion of the fibers, about 25%, that are going to the posterior portion of the VIM, right anterior to the VP, that are coming from the dentate to the posterior portion of the VIM. And you can see those dissections and also these data, also to validate the FAT1 sequence developed by Dr. Harv Akron and his team at Queen's Square. And you can see and learn more about that here. Point your camera to this QR code. So, and then here in this 3D view, you can have a understanding of the three structures. Here we have again the mammillary body and these fibers going up here, the mammillothalamic tract, going to the anterior nucleus of the thalamus. You have the CMPF, the centromedian parafascicular nucleus, that is connected to the cingulum anteriorly here, to the prefrontal area, and also to the occipital lobe. And also we have some fibers going inferiorly here to the abenula nucleus and to the dorsal nucleus of the half. And here we have some fibers right superior to it. We have the posterior fibers of the posterior commissure. So you see how broad are the connections of the CMPF. And this, it's also helpful when you have the F Gator sequence. And here we have the CM. In right lateral to that, we have VC, and then VIM, and then VO. And for these patients with writer's cramp, it was very helpful because then we got to stimulate the GPI and also the anterior portion of the VIM to get some fibers of the DRT that were going up, and then we got to improve the dystonic tremor that she had. And also, this is very important to help essential tremor patients. And this patient could not even hold his grandchild, and after surgery, he went back to holding her. And so it's really life changing to these patients, and it's the main reason why we are doing all this research that I spent 2 years at Dr. Rotten's lab, but it's still, that's why I'm in Austria to help in the course and also to study more neuroanatomy to answer some questions. I want to thank my mentors, Dr. Rotten, Dr. Kellyfoot. Dr. Rotten passed away when I was in Florida in 2016, and Dr. Kellyfoot is still there, a great functional neurosurgeon. Here is Dr. José Oswaldo de Oliveira from Brazil, my mentor. My team in Brazil, my team, the team in Queen's Square, and also in Harvard that I've been collaborating, Mayo Clinic, and Austria. So I thank all of you. And if you have any questions, I'll be more than happy to answer. You can just shoot me an email or get connected with me through social media. I wish you success, a successful event. Bye-bye.
So this concludes our session, but before we go, I want to thank Boris and John, my co-chairs, for organizing this session, all of our speakers, Aileen, Michael, Tobias, and Vanessa, and feel free to connect with them directly if you have any more questions. And finally, thank you for attending and for your attention.